Good morning, good afternoon, and good evening, everyone, from wherever you're joining us. And welcome to today's webinar titled Control of Muscle Glucose Uptake in Vivo, Thinking Outside the Myocyte, featuring Dr. David Wasserman, Professor of Molecular Physiology and Biophysics and A.M. Lyle Chair of Biomedical Sciences at Vanderbilt University. Today, he will help you all gain a more complete understanding of muscle glucose uptake through consideration of the integration of physiological systems that control this process. I wanted to take this moment to thank our partners at the American Physiological Society and the sponsor of this event, Kent Scientific, for making it all possible. Without any further delay, let's get into it. And I'm going to welcome David. Hi.
Hi, thank you, Sydney.
Go ahead and take it away. Thanks.
Okay. This is my intro slide, and I'm going to use this to introduce the topic. Muscle glucose uptake requires 3 serial steps. This is intuitively obvious that the glucose has to be delivered to muscle. Transport of glucose then must be across the muscle membrane, upon which time it's phosphorylated within the cell itself. Now, I'm going to focus on the extracellular, the extramyocellular delivery of glucose, but I also am going to emphasize that there are changes within the extracellular space, the local muscle environment, which affects mechanisms for glucose uptake within the cell. I would also like to add, this slide is drawn to show glucose delivery, but the delivery of insulin is an equally is an important process for insulin-stimulated muscle glucose uptake, obviously, and in fact faces greater barriers to getting to the muscle cell. So today I'm going to talk about the microcirculatory barriers to insulin-stimulated muscle glucose uptake, and I'm going to talk about a relatively unexplored area, and that is the extracellular matrix, the ECM, integrin receptor axis, as a contributor to muscle insulin action and muscle insulin resistance. So just by way of introduction, the vascular delivery to myocytes can be split into 3 components. Microvascular flow is controlled by vascular reactivity. So in the case of insulin, it activates an endothelial-dependent mechanism via eNOS, resulting in the production of NO and the activation of the increase in cyclic GMP in smooth muscle, which causes vasodilation. Capillary density is a key determinant because this determines the surface area of exchange. So capillary density is a critical component. And finally, the molecules exchange between capillary lumen and interstitial space, and it does so by crossing a high resistance endothelial barrier here. Now, there are some molecules like glucose who can get out between little gaps in the cells. On the other hand, proteins, such as insulin, may be too big to get out through the gaps and are passed through the cell by transcytosis. So this slide shows insulin, of course, is the major hormone that stimulates glucose uptake in skeletal muscle and some other tissues. And this shows insulin. This is an old study by Mark Laxo and Alan Barron that looked at the insulin dose-response relationship of whole body glucose uptake and then leg blood flow. Now, as you can see, there's a dose-response relationship between whole body glucose uptake and insulin. And there's a right shift with obesity and then with type 2 diabetes. And leg blood flow shows the same rightward shift. So there's clearly a correlation. The causality and how this links to whole body glucose uptake is harder to know. But clearly the endothelial dependent vasodilation is impaired in insulin resistant states. this slide, this is arterial insulin, this red line right here, and this is the percent steady state responses. And what you can see is that arterial insulin rises very rapidly in the blood. But in terms of getting into the interstitial space, the site of action, it rises quite slowly and maybe reaches a steady state in a somewhat longer fashion. Now, the interstitial space, of course, is what bathes the parenchymal cells, and you can see the glucose disposal runs in line with the interstitial insulin and not the arterial insulin. So there's a barrier to insulin getting across here. And this is due, there are two determinants of this barrier, and that is the capillary density, the surface area for exchange, and then the permeability of those endothelial cells for insulin. So let me just show you the capillary endothelial barrier to insulin. This is the arterial levels in lean and obese subjects, rises rapidly. And again, you see in the interstitial fluid, insulin rises more slowly. But in obese individuals, there's an even greater lag, suggesting that the onset of insulin action will be delayed in these subjects. So a site of potential insulin resistance. So I'm going to summarize some studies we did. This slide will summarize about 5 years of work from two outstanding postdocs at the time. And what they found is that insulin crosses capillary endothelium by a vesicle-mediated fluid phase transport mechanism. And this is where the lag occurs. So in mice, these were done in mice using intravital microscopy, isoflurane anesthetic. And it took about 10 minutes to equilibrate with the peri-interstitial space. That is the space immediately outside the vessel. And not the full interstitial space, but just getting across. It took much longer to get in to equilibrate with the entire interstitial space. And so what this means is that insulin is dispersion limited. Okay, how easily it gets out from the capillary lumen. Now, glucose exits through intracellular gaps, and it equilibrates with the peri-interstitial space within seconds. It's approximately 3 seconds to get to equilibration with the peri-interstitial space. And so this is perfusion limited. And the implications of this are important because what it means is that you can increase perfusion, but not, that is the flow rate, without increasing insulin delivery, but you do increase glucose delivery. On the other hand, insulin is more reliant on permeability of the endothelial cells to insulin and the activity of the vesicular transport. Now, insulin-resistant DIO, diet-induced obese mice, and DBDB mice, which lack the leptin receptor and get very fat, have dysmorphic endothelial ultrastructure and mildly impaired transendothelial insulin efflux. And this could contribute to the insulin resistance. Now, in the severely resistant DBDB mice, we have a reduced number of capillaries. And this is key because it limits the surface area for exchange of both glucose and insulin. Now, I want to spend a few minutes on a method because this is going to be the centerpiece of much of the data I'll be presenting. So in order to test insulin action in mice, we used the hyperinsulinemic euglycemic clamp. This is an example of the clamp method. 1 week before an experiment, through a ventral incision, we catheterize a carotid artery and a jugular vein. The free ends of the catheters are tunneled to the nape of the neck where they're attached to extensions. 1 week later, they've regained weight, normal behavioral activity, eating, drinking. We attach them to these free ends to extensions for arterial sampling and for venous infusions. Now, for a insulin clamp, we infuse insulin, of course. And if you infuse insulin without glucose, the mouse gets, or any species would get hypoglycemic. Here, for a clamp, we infuse glucose at a variable rate based on feedback from frequent samples. Now, we can also combine tracers. In the example I'm showing you here, we use tritiated glucose and 14C2-deoxyglucose. Now, in most of the slides I will be showing you, the readout is going to come from here. So the 14C2-deoxyglucose gives a tissue-specific readout of glucose uptake. And when you inject 2-deoxyglucose, it's transported into the cell, it's phosphorylated in the cell, but not metabolized further. So what we can do is measure the rate of accumulation of the phosphorylated 2-deoxyglucose to get an index of glucose uptake. And this slide just shows what a day in the life of a hyperinsulinemic euglycemic clamp. Animals are fasted for 5 hours prior to the beginning of the clamp. We have an equilibration for the tritiated glucose infusion. We then, starting after the equilibration period, we then give insulin. This dose is what we generally use for studies that look at muscle because it's right in the steep part of the insulin glucose uptake dose response curve. It gives high physiological levels you might see after a high carb meal. Now, and of course, we infuse variable glucose, and we include tritiated glucose with that infusion to keep specific activity, that is the ratio of radioactive to cold glucose, in a steady state. And this makes our computational end stronger. Now, importantly, we give 2-deoxyglucose at 78 minutes and then sample frequently for 2-deoxyglucose, get a decay curve. We then excise tissue after sacrifice at 120 minutes and then measure in a tissue-specific way the amount of phosphorylated 2-deoxyglucose. So using this method, we studied the effect. We used a model that results in a a decrease in muscle capillaries. So these are lean, otherwise healthy mice, but we knocked out VEGF specifically in the muscle. And what that does is it results in normal circulating VEGF levels, which is an important angiogenic hormone. But locally, it results in a 50% reduction in muscle capillaries. And you can see that with this 50% reduction, there's approximately a 50% reduction in the index of glucose uptake obtained using 2-deoxyglucose. Now, is there a muscle effect? So when we studied the same, a separate cohort in isolated muscle, the effect was gone. Okay, so there's basal 2-deoxyglucose, insulin stimulated, and soleus extensor digitorum longus. And as you can see, they're both unaffected, suggesting that it's an extramyocellular effect. So the capillary density is clearly important. We were interested in knowing whether the increased, or whether in DIO mice, we could correct the insulin resistance seen in these animals by treating them with a vasodilator. In this case, we use sildenafil treatment. And these studies were done by Julio Ayala. And with vehicle, these mice, with sildenafil, you can see that the insulin resistance was at least partially corrected, suggesting that there may be a vasodilator effect. However, that sildenafil acts rapidly. This is the a name for Viagra. So it acts rapidly. And yet, when we give it acutely, it had no effect. And this was surprising to us. We expected an acute sildenafil infusion to have a positive effect on glucose, to be able to affect glucose uptake similarly. So we then did the same thing with relaxin, another vasodilator. Now, this was an intervention study. We took DIO mice and treated them for 3 weeks with relaxin. And this too increased muscle glucose uptake. This is gastroc and superficial vastus lateralis. Open bar is relaxin, dark is vehicle. And this too increased glucose uptake. The 3 weeks of treatment. Now, these studies were done by Jeff Bonner, whose picture disappeared. Now, in isolated muscle fibers, the effect is gone, suggesting that this is it's because of an extramyocellular mechanism, presumably due to an increase in flow. However, again, acute relaxin had no effect. So we just give it the day of the experiment during the insulin clamp. It had no effect. So this led us to 2 observations and a question. Studies of a variety of model systems and humans show that an extramyocellular barrier to glucose uptake exists in healthy and insulin-resistant models. Okay. But chronic but not acute treatment with vasodilators increases insulin action in DIO mice. So why is it that the acute is ineffective but the chronic works? And so we asked the question, are adaptations to the extracellular matrix coupled to insulin action? We looked at things that could possibly be changing over time and asked, is there an adaptation at this site that affects the local environment? Now, here we showed, these are 18-week-old mice. The DIOs were treated for 12 weeks with, were given a high-fat diet and made a diet-induced obese. And this hydroxyproline is a marker of extracellular matrix protein. And you can see there's a decisive increase in DIO mice. We selected collagens 3 and collagens 4 mainly because of their abundance in muscle. Collagen 4 is also a key basement membrane protein. And you can see both of these are increased as well. So we took this to mean that the ECM is indeed increased by muscle proteins, or in DIO mice. So this increase occurs for 2 reasons. There's an increase in the transcription of the collagen chains, but also there's a reduction in MMP9, which is one of the primary enzymes which breaks down matrix proteins, collagen specifically. So we started with this, we worked with this hypothesis. So supply-demand results in a metabolic stress, and this may be, there are a number of hypotheses for what this metabolic stress may be, lipid metabolites or oxygen-free radicals. And this leads to inflammation, which we hypothesize results in ECM excess. And here's where the hypothesis is, where we think signals cell surface receptors called integrins. And this contributes to the insulin resistance. So this is a slide, a scheme that shows the extracellular matrix. Here's the cell membrane and the cytosol. And with a high, in DIO mice and in humans, we know, if there's a gain in weight, there's an increase in ECM proteins. Now, these proteins are not simply physical barriers. They're ligands for what's called the integrin receptors on the cell surface. And every cell expresses integrin receptors. And our question is, do ligands activate integrin receptors in DIO mice, thereby contributing to insulin resistance? Now, just a brief introduction. I'm going to focus, there are 18 different alpha subunits and 8 beta subunits in the heterodimeric integrin receptors. So there's an alpha and beta. The alpha subunit conveys specificity of ligand binding, whereas the betas have a tail that exude into the cytoplasm and signal to the cell adhesion proteins. So beta 1 and alpha 1 and alpha 2 and 10 and 11 are collagen binding proteins. And I'm going to focus on beta 1 and then the alpha 1 and alpha 1 and alpha 2. Actually, I'm going to focus on alpha 2. And I'll tell you right now, alpha 1 subunit deletion had no effect. So I'm going to focus on alpha 2 rather. So the first thing we asked is, does the integrin receptor and changes in the matrix affect the blood flow and capillary density? Now, deletion of integrin beta 1 from the endothelial cell is embryonic lethal. And in fact, an inducible knockout after about 4 weeks becomes lethal as well. Now, we worked really hard to find the sweet spot where we could induce it, knock it out, or knock it down rather, and the mouse would still be healthy. So we found 10 days after tamoxifen injection, we had just a modest drop in a knockdown in integrin beta 1. But the mice gained weight normally. Their exercise tolerance was normal on an exercise stress test. Now, this knockdown resulted in a decrease in the capillary marker CD31, implying that capillary density is less. And you can see the merger of the two images showed a similar reduction. So the knockout on the endothelial cell these are lean mice, took otherwise lean mice and caused capillary rarefaction. So we used intravital microscopy, and I'm not going to go into great detail. The nose cone's not shown here. We used isoflurane. We have a jugular vein catheterized, and we use that for injection of fluorescent probes and any other reagent we want to put in. Now we have them, the body temperature is maintained with a thermostatic heat blanket, and we superfuse the muscle with oxygenated saline and at body temperature. Now, we use confocal microscopy and then take the reflected images and divide it up to look at the specific wavelength of our region of interest, of our molecules of interest. What we found is that knocking out the endothelial cell, or knocking down the endothelial beta 1 subunit by about 30% resulted in a roughly similar fall in mean flow velocity and a decrease in the percent capillaries that were perfused. So it had not only an effect on capillary density, but an effect on the flow, on the rate of blood flow through the capillaries. Now, what did this all mean for insulin resistance? So this is the same mouse model, and this is 10 days post tamoxifen, an inducible endothelial integrin beta 1 subunit knockdown. That's a mouthful, and it results in insulin resistance. So here's the wild type, or the non-Cre control. Both groups had tamoxifen, but this one lacked the Cre. And you could see that glucose infusion rate was reduced in the knockdown. And this was due to an increase in glucose disappearance and generally, for the most part, a decrease in muscle glucose in tissue glucose uptake. Here's muscle glucose uptake, and then adipose tissue glucose uptake was impaired. So a primary defect in the endothelial cells is a factor in and can potentially cause insulin resistance. This phenotype that we observe is not dissimilar to what you might see in a type 2 diabetic subject or a DB/DB mouse. So the conclusion is the deletion of the endothelial cell integrin beta-1 subunit, and probably knockdown would be a better word, creates a type 2 diabetes phenotype in otherwise healthy, lean mice. So let me turn now to how the matrix may affect the muscle itself. So the first thing we did was a freezer study where we took mice that we had demonstrated had increased insulin sensitivity, either through genetic manipulation or by pharmacological manipulation. Okay, and this would be the PDE5A inhibitor, sildenafil, which I showed you a bit of earlier, and then the genetic expression of mitochondrial catalase. These were studies done by Li Kang. And what I want to show you here, the mitocatalase expression is shown in this first panel here. And this is F480 and TNF alpha, both markers of inflammation. The open circle are lean, dark, or DIO. The markers for inflammation are increased with DIO mice, as you might expect. And this is from muscle tissue. Now, when we made the animal more insulin sensitive with catalase expression in the mitochondria, we normalized these inflammatory markers. Same with sildenafil. Now, these are both groups are dark, which means they were both on a high-fat diet. These were vehicle treated, and this is sildenafil treated. And these two had a decrease in inflammatory markers. And this corresponded to decreases in the two collagens 3 and collagen 4. So increasing insulin sensitivity, decreasing inflammation, led to decreases in these key extracellular matrix protein. Sildenafil did the same thing, decreased extracellular matrix proteins. Now, in looking at these mice, here we looked at capillary endothelial cell markers, CD31 von Willebrand factor, in the catalase expressing and mitochondrial catalase expressing mice and sildenafil treated mice. And what you can see here that's notable is that the capillary markers are dramatically increased. So not only is there less extracellular matrix, but there's more capillary proliferation. So it seems based on these histological markers, which in and of effect would improve delivery of insulin and glucose to the insulin sensitive tissue. So the working hypothesis we can add to this, and we know that events that correct insulin resistance, or at least partially correct it, whether they be genetic or pharmacological, decrease inflammation and the excess ECM. The question is now, what do they do with the integrin? Do they have actions through the integrin signaling protein? So this slide is very complicated, but I'm going to try to simplify it. I told you before, the integrin receptors are heterodimers, an alpha and beta subunit. Now, I've highlighted the cell adhesome, by the way. This is a minimal example. There are about 150 different proteins that can be comprised as cell adhesion proteins. I'm highlighting the IPP complex, ILK, pinch parvin. These are highly conserved scaffold / adapter proteins that are expressed in all cells and retained in a vast number of species, at least their homologs are. And the darker proteins are ones that I put in for because they're there. But I'm going to focus on these and the integrin receptors. Specifically, we studied three different integrin receptors and alpha subunits. Alpha 2 subunit had an effect, and that's what I'm going to show you. Okay. So this is the hyperinsulinemic euglycemic clamp in integrin alpha 2 subunit knockouts and how it affects muscle insulin action. Here are lean mice. In lean mice, knocking out this integrin had no effect on the glucose infusion rate required to maintain euglycemia or glucose uptake. In DIO mice, however, glucose infusion rate at a steady state was almost doubled, and there was about a 5-fold increase in the index of muscle glucose uptake obtained with 2DG. So the fact that knocking it out has a profound effect in increasing insulin action suggests that its presence is required for the insulin resistance. So I want to then turn to, we went down the tree because we're looking for mechanism, how the integrins work. And we looked at the center of the IPP complex, the integrin link kinase. And the name is confusing because even though it's named a kinase, the kinase consensus site is actually a binding site in vivo and doesn't seem to have kinase activity. Now, we explored and focused on this enzyme. Let me elaborate a little bit on this IPP complex because for simplicity, I divided up into two parts. The pinch side, which works on a tyrosine receptor signaling, insulin signaling, and the parvin, which orchestrates the cytoskeleton side. Okay, and ILK can affect either one of these. So back to ILK. And what you can see is the phenotype was not unlike the alpha 2 receptor subunit knockout. No effect in lean mice of glucose infusion rate or glucose uptake. But if you look at the glucose infusion rate in DIO mice, you see a marked increase in glucose infusion rate, and the glucose uptake increases by about 3-fold. So this is a very marked effect of knocking out the integrin-like kinase. Again, the idea that if you knock it out, it improves insulin action means that its presence is required for insulin resistance. So what I didn't show is deletion of muscle integrin and ILK in DIO mice improves insulin signaling. They increased the number of capillaries in the muscle, but the ECM protein content remains high. So what that means is that it's not acting by reducing the ECM proteins, but rather by through the interaction as ligands with the integrins. Now, and none of these effects occur in lean mice. Okay, so it seems to be specific to DIO mice in the inflammatory state with the increased ECM. So this takes us a step further, and we can say that integrin signaling may in fact be a part of the pathogenesis of insulin resistance. And this is from the integrin alpha 2 beta 1 receptor and the ILK knockout mice. So back to this busy scheme. So we tested pinch, and we didn't find a phenotype in pinch. They responded normally to insulin clamps, whether it was present or not. And I'm not going to show that data. I hope you'll take my word for it. We then went to alpha-parvin, which was very unique. So alpha-parvin orchestrates a very complicated reorganization of the cytoskeleton. And it does so through row kinases, cofilin, and various other regulatory proteins. And this is important. These processes are important for translocation, for migration of proteins to the mitochondria, mitochondrial structure function. So this is potentially a high impact protein and deletion. And what we saw was absolutely fascinating, because in these mice, it was the lean that had a decrease in muscle glucose uptake. This is gastrocnemius and superficial vastus lateralis. And you'd see that deleting parvin in otherwise healthy lean mice had a marked decrease in glucose uptake, and the DIOs had no effect. Now, paradoxically, we have a small increase in glycogen here. And I'd be happy to answer questions about that if it comes up, if people have questions. I won't get into it right now, but that was fascinating. And despite all the problems, the impaired glucose uptake. But again, in DIO mice, no phenotype, at least in terms of the variables we measured. Now, this slide, if you look closely, the green is GLUT4. CAV3 is a membrane marker. So what you want to look for, the co-localized GLUT4 CAV3, so the GLUT4 on the plasma membrane, F-actin as a cytoskeletal marker, and then DAPI just identifies cells, cell nuclei. So this is a wild type mice, and these are the blow-ups of these two regions here. So what you'll see is an abundance of the white F-actin, and you can see abundance of green, the GLUT4, co-localizing on the membrane. And that's true on both blow-ups. Now go to the knockout mouse, and you can see that here's the general slide, and here's the blow-up. And you can see that the F-actin is very sparse along the membrane, which is the CAV3 marker, delineates that. And except for, we have a cluster of GLUT4 here, but otherwise, the amount of F-actin and GLUT4 is greatly diminished with the parvin knockout, suggesting that cytoskeletal arrangement is dependent on this cell adhesion protein. And this is a quantification of GLUT4 co-localized with CAV3. And as you can see, there's about a 50% reduction when we put all the slides together. Now, a second area of cellular disorganization was in response to insulin. We saw that wild-type mice had more hexokinase 2 localized to the mitochondria. In the MPAR knockout, we saw no such relationship of HK2 to mitochondria. It was more cytosolic. And here are the Western blots for a small group of those. So integrin signaling regulates insulin-stimulated muscle glucose uptake. So integrin alpha-2 subunit and ILK are required for muscle insulin resistance in DIO mice. And this is evident by the fact if you knock them out, they get more insulin sensitive. Now, alpha-parvin is required for translocation of GLUT4 to the muscle membrane, localization of HK to the mitochondrial membrane, and I didn't show this, but mitochondrial function as well. Alpha-parvin does not impair the insulin signaling pathway. So we speculate that the effects of integrin alpha 2 subunit and ILK act, at least in part, by disengaging or deactivating alpha parvin. So, and this scheme I've drawn sort of a ghost image of alpha parvin, and if you'll notice, I've decreased the cytoskeletal arrows on this slide. And the hypothesis that I'm going to end with a hypothesis, which is that integrin ILK activation by DIO disengages alpha parvin, impairing cytoskeletal function. And this is phenocopied by an alpha parvin knockdown, to be precise. Now, with this, I wanted to end with this because we're at the tip of the iceberg. And the cell adhesome and the integrin receptors are very complicated. There are many different integrin receptors. And to isolate on just a couple subunits is tip of the iceberg stuff. There are many cell adhesion proteins that remain to be explored in terms of their effect on metabolism and cytoskeletal organization. So there's much to do in this field. And I finally just want to thank the people who participated in this work in the laboratory and previous members who participated in this project. And my collaborators, Ambra Pozzi and Roy Zent, who introduced me to the area of integrins, and my longtime colleagues, Owen McGinnis and Julio Ayala, who always have some wise words to say during as the work progresses. And with that, I'd like to thank you and open the floor for questions.
Fantastic presentation, David. Thank you so much. Our first question is, the soleus in mice is mostly type 1 oxidative fibers, whereas the gas is predominantly type 2 glycolytic fibers. Are you not surprised to see similar glucose uptake by both?
So the soleus, yeah, I'd have to go back to the slide. Soleus glucose uptake is much higher. And I'm having trouble referring to the slide. We usually have to plot them on different scales. Now, we don't always show, in fact, we usually don't show now. We've stopped putting the soleus as part of the scheme simply because it's a very small muscle. And I became concerned that when plotted on the same scale, it was over-representing the muscle phenotype. So it's only like a 10 milligram, well, it's very small, it's about 10 milligrams in the mouse. And yes, it should be, and I apologize on my slides if it didn't reflect that, but it should be much, much higher. It's closer, it's about halfway in its glucose uptake between the gastroc and the diaphragm. Now, the gastroc we use is usually been a mixed lateral medial. So it should be more representative than strictly type 2. Did that answer a question?
I think so.
Okay.
I suppose
but the questioner is correct. I mean, the soleus is much higher. And I often don't include it simply because it's so much higher that it's not representative of muscle, of the bulk of muscle.
Okay. Let's see. In addition to skeletal muscle, glucose uptake changes. Is there any effect in the heart muscle that you know of? Yeah,
I think the heart may even be more profound in that way. So the heart is more prone to fibrosis. Okay, so more ECM remodeling than the skeletal muscle is, for example. So myocardial fibrosis, liver fibrosis. We study the liver quite extensively, but there's a field that looks at myocardium as well. And I would expect that to be a very important part of myocardial, of heart disease, really.
Okay. Thank you for sharing that. The difference in glucose uptake after B1 subunit knockout in skeletal muscle is very interesting. Do you think glucose uptake is muscle specific even at rest?
So we didn't do, okay, we did B1 in endothelial cells and not directly in muscle. Now, I have to say, my colleague in New York did, Jeff Pessin at Einstein and his colleagues did a B1 knockout in mice, in muscle. And I've been trying to get my head around his studies. His phenotype, by the way, looks more like the alpha parvin phenotype that I showed than the alpha 2 subunit receptor. So this gets into a very complex interaction between alpha and beta subunit pairings. Now, I have completely forgotten the question.
So the difference in glucose uptake after B1 subunit knockout in skeletal muscle is very interesting. Do you think glucose uptake is muscle specific even at rest?
Okay, resting, I think it may be, but the resting glucose uptake tends to be rather small and hard to pick up in vivo or even in vitro, because I think the same mechanisms may apply. Of course, translocation at rest is not so prevalent. You need a stimulus like exercise or insulin. But it's tough to say because at rest, it's very hard to resolve a difference just because of the low glucose uptake rates. So it's a good question. I just can't pick that apart.
All right, that's okay. Maybe something that you can look into in the future.
Yeah.
Okay, so do you think the defect in adipose tissue glucose uptake with B1 integrin deletion is more important than muscle glucose uptake in obese mice and humans? And would you think this would be opposite in lean mice or people?
Okay, B1. Okay, so we're going back to the B1, and I didn't actually look at B1. That was in the endothelial cells. But yes, right, B1 in endothelial cells show, and I do think that's very important. Ultimately, that, there's a lot of work that shows the ability of the adipose tissue, their perfusion by capillaries, their ability to store fat is a very good protective mechanism in preventing metabolic disease. Now, we didn't look at adipose tissue. One of my colleagues on those slides, she was a postdoc at Lee Kang, and she's an excellent scientist. And when she went to be an independent investigator, I said, Lee, do the adipose tissue stuff. And so she's been working away and has shown done some excellent work on the integrins and CD44 receptors and other things on adipose tissue. Now, the field as a whole would suggest that anything that would impair the function of an adipocyte, whether it's a decrease in capillaries to it, the ability to get oxygen, the ability to store fat, is going to cause insulin resistance, because the fat that can't be restored in adipocytes has to be stored elsewhere. And that could be liver, muscle, heart. And these lipid metabolites that collect may be the cause of insulin resistance, or at least a cause of insulin resistance. So I think, yes, it's the adipocyte may be really quite central to the whole thing.
Okay, very interesting. And maybe we need to ask your colleague to come next time for a follow-up webinar.
Yeah, well, she's about 8 hours difference, but she'd be.
That's great. Okay, is there any data showing an increased expression of ILK or integrin alpha 2 in diabetic population? Would this not be a good pharmacological target?
Yeah, we don't see an increase in liver. We've seen increases. Okay, but keeping with muscle, we haven't seen an increase. And the reason we're working downstream on this, and progressively, this is part of a series where we're going down the integrin signaling pathway, is the concern about is that these proteins do not have catalytic sites. They don't have enzyme activities to inhibit. So we'd be more reliant on disrupting protein interactions, which is a harder tool, is a harder therapeutic, makes it a harder therapeutic target. So, but if we get down further in the cell adhesome, where you're looking at rho kinases and actinin and other proteins, or cofilin, for example, there may be ways to enhance, and ultimately, we hope that there will be something in there that may be druggable and improve the ability to improve the organization of the cell, the ability to move proteins into the proper compartments, like on the membrane when they're needed, in the mitochondria when they're needed. And the other thing, which I didn't get into, but the cytoskeleton is key for mitochondrial structure function. So these specific proteins may be hard to drug, but it may be that downstream, the proteins which actually have catalytic activity, the enzymes, might be viable targets. And there are drugs, particularly in cancer research, that are used.
Okay. Great response. Thank you. We do have just a few minutes left, so we'll try to get to one, maybe two more questions. And then I know everybody would love their question answered. So we will be sending all the questions to Dr. Wasserman to provide a Q&A report. So you can continue to submit your questions, and we will pass them on to him. Okay, David. Which effect does HIF induced by hypoxic conditioning have on the capillary function and insulin resistance?
You know, I wish can I take I am not, I'd have to look that one up. Okay, I apologize.
That's okay. Yeah, well, I'll pass these on to you, and yeah, you can look more into it, and then
the stump the panel person one.
Good job. Okay, we'll move to our next one. Does altering integrin receptor signaling affect exercise stimulated muscle glucose uptake in vivo, or contraction stimulated muscle glucose uptake ex vivo?
Probably both. So I can tell you in vivo, I did not discuss this, but the muscle knockouts have decreased exercise tolerance on an exercise stress test. So that would suggest that it is impacting on the organization of sarcomeres or contractility or possibly calcium release. So these mice, even though they're normal weight, activity in the cage is normal, if we put them on an exercise treadmill and do an incremental exercise test, their tolerance is a bit lower. So,
okay, interesting. Let's get to one final question for our time today. So, is translocation of GLUT4 to membrane by contractile activity affected in alpha parvin knockouts? Can you dissociate insulin versus contractile activity effects on GLUT4 translocation as well as glucose uptake?
Yeah, so we didn't do that, but that's a good question. I expect that we can, because I think this contraction exercise requires cytoskeletal regulation, just like insulin does. And even though the mechanisms to activate the translocation are different, ultimately it does come back to the ability to reorganize the cell so that more GLUT4 is in the membrane. So I expect that they would both be characterized by this defect. And but we haven't done the study. Yeah, we can do it before we go home today.
Yeah. Why not? Who can stay on with us?
Yeah.
Okay, Dr. Wasserman, thank you so much for your time today. I think there's lots of comments in here that I'll be passing on to you that also agree. It was a great talk, and yeah, it was a pleasure to have you join us.
Well, thank you very much for asking me, and thank you very much to the audience for sitting through this, for listening to me.
In closing, we hope you enjoyed this Inside Scientific webinar produced in partnership with the American Physiological Society and sponsored by Kent Scientific, and I hope you enjoy the rest of your day.